Well, this afternoon, we're excited to have a conversation with Andrew Tsourkas. He's the founder of AlphaThera and a professor in bioengineering at Penn. And Andrew, one of the things that impressed me, I'd like to hear about your path to Penn. Man, you hit a number of really well-recognized institutions. How the heck did you get into this? Because your story is a great story.
Well, first, thanks for having me. Yeah, it's been an interesting path, to say the least. So I started my undergraduate degree in mechanical engineering up at Cornell University, and really enjoyed my time there. It was amazing. And even though I was in mechanical engineering, my focus was in biomedical engineering, so in particular biomechanics. So that's a far cry from what I'm doing now, but I think it got me introduced into the field. From there, I went to start my PhD at John Hopkins University and switch from kind of macroscale biomechanics, things like knee replacements, and started studying things at the cellular molecular level. About halfway in, I went down to the Georgia Tech Emory joint program. I moved with my advisor. So that was not necessarily my decision, but my decision to go with him to that program. And then next ended up at Harvard University at Mass General Hospital. And that's kind of the first time I really got into the field that I'm in now, which is molecular imaging and drug delivery. I worked in a big lab there under the direction of Ralph Weisslieder, and then was very fortunate enough to have my first faculty position here at the University of Pennsylvania in bioengineering. So I've been fortunate to be to so many great places and really meet some great people along the way.
So you talked to us a little bit about the work that you were doing in your lab, and you were headed in one direction, and all of a sudden, you kind of discovered some things that weren't really the focus of what you were doing. Can you?
Yeah, no, that's right. Yeah, thanks. So our lab historically has been a nanoparticle or nanomedicine lab. So nanoparticles, just briefly, are typically very small particles that are used to package drugs to improve their delivery in patients, so to help drugs get to the desired location. And one of the ways we and others have done that is to attach things like antibodies or other proteins that can direct them to a specific site. And that site can be a cancerous site or some other disease like atherosclerosis. And one of the challenges that we always faced was attaching these antibodies to these nanoparticles, where it would vary from hand to hand. So one person may have more success than the other. But ultimately, we were even lucky if we could get 10% of these antibodies onto the nanoparticles. And to be honest, usually it was on the order of about 1%. And when we went through the entire process, we would end up throwing away all those unattached antibodies. So we'd throw away 90, 95, 99% of the reagents that we were working with. As you can imagine, it's very wasteful, but it also prevented us from working with a lot of other people at Penn. So they would be very interested in our nanoparticles and tell us they wanted to target their disease using their antibodies, and the amount of antibody we would need because of this wasteful process would be just, an order of magnitude too much. It was just way too much than they could provide. So it really forced us to kind of step back and see if we can come up with a solution. Is there any simple, easy way to efficiently attach things like antibodies onto nanoparticles? We were fortunate we were able to come up with a few solutions that not only improve the efficiency up to as much as 100-fold now, but also allow us to control the orientation of those proteins on the surface. So before, those proteins could be anchored to our nanoparticle or any surface, but they would be randomly oriented, and many of them would not be fully functional. But now we've also gotten control over that. So we kind of improved the efficiency, and now we have what are called these site-specific methods that give us control of the orientation of these proteins as well. And that's had a dramatic effect on how well that we can target the nanoparticles, on the conservation of materials. And as you said, it also allowed us to pursue new directions. So now in many of the things we're doing, we don't necessarily need nanoparticles, but we can attach other things to antibodies, including drugs or imaging agents. And it's also facilitated a lot of other other types of experiments that we and others do in the lab would have called immunoassays, which is really what's used in diagnostics. So anything from a pregnancy test all the way through a cancer diagnostic test, they use antibodies, and our ability to now attach these antibodies with a lot more control of the surfaces improves those assays as well.
So you live the demanding life of a professor and a principal investigator in a lab, and the question becomes, well, how did you have time to do what you needed to do to shift direction and to start to innovate and create a company? I think, if I understand correctly, you got a golden break in that department. Can we talk about that for a sec?
No, of course. Yeah, that's exactly right. Run an academic lab is extremely time consuming. As you can imagine, we have teaching responsibilities, advising responsibilities, and that's on top of our research, which is where we probably spend most of our time. But it just turned out that when we developed this technology, it was around the time where I had saved up enough time for a sabbatical. So otherwise, I think it would have been very difficult to really learn about entrepreneurship and what's necessary to start up a company. So I was fortunate enough that I had earned a full year of sabbatical at the same time that I started the company. So that gave me a full year where I didn't have teaching and advising responsibilities. I was still running my academic research lab, of course. But it gave me the extra free time I needed to really put into to kind of learn the streets and figure out what was necessary to really get a startup company off the ground, and also to spend time applying for some funding for the startup, which we were fortunate enough to receive through the NIH through their SBIR program.
Well, and we will come back to the SBIR program. One of the things that I also found fascinating is to build on the theme, as luck would have it. Well, when it was time for you to hire a first employee, man, it just seems like everything conspired in your favor. Tell us about that.
Yeah, I'm still amazed at this one. So it's so whenever you form a startup, so much depends on those very first people that you hire. So it's really make or break. So if you end up with the wrong people, it can really turn a company upside down. But I get very fortunate. There's one other lab in the entire world that works on anything even similar to what we do in terms of antibody labeling. And they're in Europe. And one of the graduate students from that lab, Fei-Fan Yu, just at the time I was forming the company, had contacted me. So we were very fortunate to have our first employee, somebody that was very familiar with our technology and knew how well it worked from our own experience working on similar technologies. So I couldn't imagine hiring anybody better to come in as the first scientist to really get everything up and off the ground. So even I am still shocked at the amazing luck that we would have somebody just at that time with that experience coming in and reaching out to look to see if we were starting a company around this tech.
So you mentioned the SBIR funding, and so you found yourself, you applied, you got the money, and now you're running a company with a handful of money to work with. Tell us a little bit more about that evolution and the growth out of your institutional research lab and out into a standalone commercial lab.
Yeah, so the NIH really has an amazing program with their SBIR program. So these are funds that are really designed to translate technologies from an academic lab to a commercial entity. And the way they typically work is they will provide funding to both sides, both the company and also the academic lab. And those funds are typically used to kind of facilitate this translation of knowledge and development of technology into a commercial product. So the nice thing about the funding as well is it's typically non-dilutive. So that's always helpful, but it really kind of builds this bridge between the two. So instead of a company just having a technology dumped on its lap, it gives you the time to make sure that that it's being translated correctly. So the knowledge isn't being lost. It kind of facilitates the interactions between the two sides, just to make sure that the eventual product is working the way it should, and that really helped. And we've been fortunate enough to get a number of SBIR grants now to really not only facilitate that translation, but also increase the number of product offerings that we have through this constant communication between the lab and the company. So now, the company is starting to gain more and more independence, and eventually will be completely independent, but it really helps kind of bridge that gap that normally exists between academics and industry.
So you've been the manager of the funds, and so it's it's forced you to make some important decisions as you've grown the operation. When you went out to find lab space, Pennovation was your choice. How did you get to Pennovation? Why was that the option of choice for you? And how's it been going at Pennovation?
Yeah, it's been great. I mean, we actually love it there. So it actually, in my mind, was relatively easy choice. So it's there are a number of places we looked at early on. But as most, I think, early startup companies, we're very, very conscientious about the money we had and wanted to use it as wisely as possible. And we think that the Pennovation Center gave us the best ability to do that. We wanted to go to a place that had all the resources we need, but really didn't need much outside of that. We needed lab benches, we needed some core space in terms of refrigerators and office meeting room, but we didn't need all the bells and whistles that came with some of the other spaces that we looked at. We really needed a workman space and wanted to use our funds as efficiently as possible. So if we didn't have to use a lot of the overhead funds that we had towards space, it allowed us to use it towards other things. And that included other overhead costs, whether it be insurance or payroll or even other materials and supply costs. And we thought that was a much better use of those additional funds. So I think working at the Pennovation Center really made a lot of sense for us.
Andrew, one of the things that we've talked about a little bit, and I think is really important to share with our broader audience, is the rocking that you've done between the research piece that's occurring in the institutional lab, and then simultaneously the work that's going on, at AlphaThera in your facility, and how you've managed that, and how it's a little bit of a foot in both worlds. So I think it'd be great if we can talk about that, and you can explain a little bit how that has worked for you.
Yeah, well, it's certainly a challenge. It's a lot of work on both sides. So, but I love my academic lab, and I think the benefits of the academic lab is it really allows you to kind of explore new research areas and test new things. And I really feel like that's what the academic environment is designed to do, is really pushing the edge of new technologies and their applications. And then on the other side, the business is really about making products that everybody could use. In commercializing technologies that work and distributing to those users that it may benefit. To me, I think what really allows us is just the excitement of both sides. I mean, the excitement of having that exploratory side in my academic lab, but also, be fortunate enough to see this other side where you actually see something that came out of my lab have an impact on other people. So it's always exciting whenever we have people contact us and tell us how it can impact their work, or, make things easier or more cost effective. And I think what makes it all feasible is just that it's very interesting, and, it's very energizing. And I see the energy on both sides, both from, these young scientists and entrepreneurs I have in the company, and then also, of course, in the lab, young graduate students and postdocs who always bring a lot of ideas and energy there. So it's been fun.
One of the things that's fascinating about your flight path is that your path to commercialization is a little bit different than for a typical drug development company in having to go through the clinical trial process. And so you've really, you're moving very quickly to the commercialization and the sale of product. Let's talk a little bit about that, so that so that the audience out there knows what you're creating and what's going to be available to them. I think it's really important. You've innovated in a meaningful way, and that your product will be out there soon.
Yeah, this was a decision we made very early on. We saw the size of the barriers and the kind of the long timeline to entry if we went in as a drug development company, which we also have the ability to do, is to make things like antibody drug conjugates and other kind of therapeutics. But very early on, we saw this alternative path to make reagents that are used in almost any biological lab, which I'll explain in a minute. So we've kind of found this shortcut to revenue generation that we ultimately decided to really head towards. So it doesn't require FDA regulations or clinical translation. So the reagents themselves are really designed to make life in laboratories or in startups or even pharmaceutical companies easier during development stage. So we develop a whole array of different agents that allow for the labeling of antibodies, which I again mentioned are used in diagnostic assays, but also used in a lot of assays for basic biological research. So we simplify this process of labeling those antibodies with a lot of control, and allow people to conjugate to antibodies in a much shorter time frame, more efficiently, with more control than current agents do. We feel like this is kind of the first breakthrough in this area in really decades. It's been really a stagnant field. So we think that we have customers in a wide range of fields now, anywhere from academics to pharmaceutical industry to diagnostic companies can really kind of make use of these labeling technologies and I think really drive down their costs and also increase their performance, because there was the additional level control that wasn't there before.
Well, Andrew, it's a great story that you're telling us, and it seems like you're right at the launch stage of this whole commercial sale to prospective customers. We've really enjoyed the time that we spent with you. We've learned a lot about how this has evolved, and a slightly different path, I think, than even you anticipated. And I'm really looking forward to catching up with you in 6 to 12 months and hearing about how the development of your product and the distribution of your product. I mean, in some ways, you said what lies ahead is really a marketing and distribution problem. And so we wish you great luck with that, and look forward to hearing in a short time about your successes.
Yeah, thank you so much for having me. Yeah, I should mention briefly that we just launched products, so on AlphaThera.com. So we'll see in the next 6 months. Hopefully, we'll generate a lot of excitement, and definitely look forward to keeping you up to date on how we're doing. Thank you.
That's great. Thank you, Andrew.